Hello everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is glycolysis. This video is taken from Plant Physiology series, CBSE class 11th, part 2nd of chapter 15. Now let's start the topic. The first thing is, what is glycolysis? Glycolysis is also known as Embeden-Merhoff-Parnas pathway or EMP pathway. All the living organisms, whether aerobes or anaerobes, initiate the mechanism of respiration by breaking down glucose to pyruvic acid without the help of oxygen. This initial process that occurs in the cytosol of the cell and completes in 10 steps is called glycolysis. In Greek word, glycose means sugar and lysis means splitting. So, splitting of sugar in absence of oxygen or without the help of oxygen is known as glycolysis. The scheme of glycolysis was first described by Embeden, Otto Meyerhoff and J. Parnas and therefore often referred to as the EMP pathway. Now, the various steps involved in the pathway. Now, here you can see here this figure show the various steps in case of glycolysis. So, the first step and the first molecule is glucose. Glucose molecule is phosphorylated in the presence of ATP to form glucose 6-phosphate. The reaction is catalyzed by enzyme hexokinase. Here you can see that the enzyme names are dictated by the green words. So this hexokinase, which requires a divalent magnesium ion as a cofactor, ATP is converted to ADP in this reaction. So the first step is conversion of glucose to glucose 6-phosphate and here ATP is converted to ADP. It means one phosphate group is released. Now, after this glucose 6-phosphate is isomerized to form fructose 6-phosphate in presence of enzyme phosphoglucoisomerase. So this is a isomerism reaction, that is conversion of glucose 6-phosphate to fructose 6-phosphate in presence of enzyme glucophosphate isomerase. Now, after this phosphorylation, so here you can see that in this reaction fructose 6-phosphate combined with other phosphate group from another ATP molecule yielding fructose 1,6-bisphosphate and ADP. So here fructose is converted to fructose 1,6-bisphosphate and here also ATP is converted to ADP. So it means one phosphate group is attached to the fructose 6-phosphate to form fructose 1,6-bisphosphate. The combination is catalyzed by an enzyme phosphofructokinase. In the presence of magnesium ion and utilized energy released by separation of a phosphate group from ATP. This phosphorylation thus consumes another molecule of ATP. Now, after this is splitting. So what is splitting? Here you can see that this fructose 1,6-bisphosphate now splits into 3-carbon phosphorylated sugars and these are first one, that is glyceraldehyde 3-phosphate, which is also known as 3-PGAL, and the second is dihydroxyacetone phosphate. So the reaction is catalyzed by an enzyme aldolase and here dihydroxyacetone phosphate is converted into 3-phosphoglyceraldehyde with the aid of an enzyme phosphotriose isomerase. Now, the fate of each 3-phosphoglyceraldehyde molecule is the same and the fate of only one is discussed below. So here you can see that in case of phosphorylation and dehydration, which involves the 3 process, in phosphorylation 3-phosphoglyceraldehyde, glyceraldehyde 3-phosphate, here you can see this, combines with a phosphate group derived from inorganic phosphoric acid found in the cytosol, not from ATP, forming 1,3-diphosphoglycerate or diphosphoglyceric acid. The reaction occurs with the aid of a specific enzyme. In dehydrogenation, a pair of hydrogen atoms separates from a molecule of 3-phosphoglyceraldehyde, and their separation releases a large amount of energy. A part of this energy is stored in newly formed phosphate bond of 1,3-diphosphoglycerate, making it a high energy bond. Separation of hydrogen is catalyzed by an enzyme 3-phosphoglyceraldehyde dehydrogenase. So, the two hydrogen atoms separated from the 3-phosphoglyceraldehyde, of these, one complete hydrogen atom and one additional electron are picked up by NAD+, which gets reduced to NADH. The remaining one hydrogen proton or ions remains free in the cytosol. Now, after this, see here, the NADH, which is a high energy substance carrying the rest of the energy released by the separation of hydrogen atoms from the 3-phosphoglyceraldehyde. Energy is actually released by the transfer of electron from 3-phosphoglyceraldehyde to NAD. The NADH provides energy to convert ADP to ATP by passing its electron over the electron transmitter system if oxygen is available. So here, after this, the process is known as, here you can see that the 3-phosphoglyceraldehyde is converted to 1,3-bisphosphoglycerate, and here NADH and H plus forms. So here you can see that an enzyme is required which is known as 3-phosphoglyceraldehyde dehydrogenase. Now, after this, dephosphorylation or ATP formation starts. So before this, ATP used. After this, the ATP will be formed. So high energy phosphate group on the carbon 1 of the 1,3-diphosphoglycerate or bisphosphoglycerate is transferred to a molecule of ADP, converting it into an ATP molecule. So 1,3-diphosphoglycerate changes to 3-phosphoglycerate. Here you can see that 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. Due to loss of a phosphate group, the reaction is catalyzed by an enzyme diphosphoglycerokinase. Here you can see that the navy blue color words indicate the name of enzymes. Here, magnesium ion is needed in this reaction. Formation of ATP directly from the metabolites is known as substrate level phosphorylation. After this, isomerization occurs. It means the phosphate group on the third carbon of 3-phosphoglycerate shift to the second carbon, producing, here you can see that this 3-phosphoglycerate is converted to 2-phosphoglycerate, only shifting of the phosphate group, and this change is added by the enzyme phosphoglyceromutase or phosphoglycerate mutase. After this, dehydration occurs, and here in this process the 2-phosphoglycerate loses a water molecule in the presence of an enzyme enolase and magnesium ion and changes into phosphoenol pyruvate. The latter undergoes molecular rearrangement that transforms its phosphate group into a high energy phosphate bond. After this, dephosphorylation occurs, that means ATP formation, and here the high energy phosphate group of the phosphoenol pyruvate is transferred to a molecule of ADP with the help of an enzyme pyruvate kinase in the presence of magnesium ion and potassium ion, and this produces simple pyruvate and a molecule of ATP. So, this is the process of conversion of glucose, one molecule of glucose into the two molecules of pyruvate, and this process is known as glycolysis. Now, after this, so here you can see the steps of glycolysis and the enzymes before this steps. So here first one, that is the glucose is converted to glucose 6-phosphate, then glucose 6-phosphate is converted to fructose 6-phosphate, fructose 6-phosphate is converted to fructose 1,6-bisphosphate, and fructose 1,6-bisphosphate is split into the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And after this, one molecule of glyceraldehyde 3-phosphate form the 1,3-bisphosphoglycerate, then 3-phosphoglycerate, then 2-phosphoglycerate, then phosphoenol pyruvate, and the last one is the formation of pyruvate. So a single molecule of, here you can see the carbon structure, here a single molecule of glucose, which is a 6-carbon compound, converted into the pyruvate, which is a 3-carbon compound. So it means one glucose molecule forms 2 molecules of pyruvate. Now here you can see the glycolysis definition, equation, enzymes and the 10 steps with the diagram. Now after this, what is the end product of the glycolysis? 2 pyruvate molecules form, which moves into the mitochondrial matrix. 2 net ATP, which is usually stay in the cytoplasm to be used by the cell. 2 used in activation energy, that means glucose activation, and 4 Medven producing pyruvate, that means energy harvest. 2 NADH, which is a high energy compound that moves to electron transport chain. So in case of glycolysis, end products of glycolysis are 2 ATP, 2 molecules of pyruvic acid, 2 NADH and 2 H2O molecules. What happens to the pyruvic acid depends on the organism involved and the presence or absence of oxygen. So in glycolysis, 3 of the reactions of the glycolysis are irreversible. Pyruvate is the end product of the glycolysis in the tissues with the mitochondria. This series of 10 reactions called aerobic glycolysis sets the stages for the oxidative decarboxylation of pyruvate to acetyl coenzyme, a major fuel of the citric acid cycle. Under anaerobic condition, pyruvate can be reduced by NADH to form lactate. This happens when the rate of NADH formation exceeds the oxidative capacity of the cell, and if there is no mitochondria or cells that have insufficient oxygen. So in glycolysis, reactants are 1 glucose NAD plus and 2 ATP, and the end products are 2 pyruvate NADH plus 4 ATP. Remember, we use 2 ATP in the process, so we have a net gain of 2 ATP. Now, so this is all about the glycolysis. So in this, total gain of ATP is 2. So ATP utilization, here 2 molecules, and ATP formations, here 4 molecules. So the total 4 minus 2, it means 2 molecules of total ATP form, that means gain in the total process of glycolysis. Next upcoming video will be on the mechanism of aerobic respiration. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.